Alright, so we've got a neuron here, which is a nerve cell, and this over here is going to be a skeletal muscle cell. And we're going to talk about how they communicate and then how one thing can cause a response in something else. So if we're looking at this, if this is a neuron that's going to speak to a muscle cell, this is going to be a motor neuron, okay, because it's efferent. It's heading away from the interneuron, which could be in your spinal cord or up in your brain. So ultimately we need a ligand, some kind of neurotransmitter. If this were a sensory neuron, this could be like a chemical that's ultimately going to be the very first stimuli that causes all of the information to be sent to the central nervous system. Anyways, this is a protein, and all of this communication happens because things are the right shape. So imagine this is a key and this is a lock, and it's going to open a door. The door only opens if the key actually fits the lock. So when these bind, then we're going to open the first sodium channel. And this is a protein as well, and that's a channel that's going to allow sodium ions in. So these positive sodium ions are going to flood to the inside of the membrane through facilitated diffusion, which is going to build up a positive voltage right here. That positive voltage is called the action potential. All along here there's a resting potential that's an overall negative charge, negative voltage, because of all these negative protein anions. So we build up a positive voltage here, and the next sodium channel is voltage gated. It doesn't open because of a ligand or some type of a chemical that lands. It opens because there's a positive voltage here. So that opens and we flood in positive sodium ions, which builds up another action potential. And we are going to propagate that action potential along here. So as this action potential charges up this region, it's going to open the next channel, which causes the positive ions to flood in, which creates an action potential. This voltage opens the next channel, allows positives to flood in, and now eventually we get down the axon to where we have a calcium channel. And again, it's voltage gated as well, but when this one opens, the sodium can't rush in because the sodiums don't fit through the calcium channel, only calcium can go in. So now these positive calcium ions are going to flood in, and they are going to go to these little proteins that are holding these vesicles of neurotransmitters off of the outer cell membrane. When the calcium interacts with these proteins, they change shape, and it allows this membrane to fuse with this membrane. As they come together, they fuse, and the contents of the vesicle is going out through the synaptic cleft. And again, this is going to be like exocytosis. And at this point, these little neurotransmitters would land on the dendrite of, or not the dendrite, but on the sarcolemma, the outer membrane. There's a receptor here on the muscle cell, and that's going to cause this first one to open. Sodium ions rush in, and we are going to send this impulse, propagate the action potential down this T tubule. And as we continue traveling here, it keeps going, and we keep on flooding these ions in. And this voltage will eventually cause a protein here from the T tubule to be removed from this sarcoplasmic reticulum. It's an endoplasmic reticulum that's all full of calcium ions. And when that protein is removed, these guys are going to flood out of that protein, that calcium channel, and they are going to flood to the skeletal muscle filaments. So these filaments, the thick ones are myosin, and they have these little oars hanging off of them. And then the thin ones are the actin. And on these actin, there is active sites where these myosin can ratchet, but they won't attach until the tropomyosin is moved off of the active site. So right now it's preventing contraction until calcium lands on the troponin, which moves the tropomyosin off of the active sites, and once the active sites are exposed, then the myosin can attach. And when it attaches, we have to use ATP now. We have to use some energy. So losing a phosphate will allow the cross bridge to form, and as the cross bridge forms, it's attached. When we lose the ADP, now it's going to ratchet, and as it pulls, it's going to shorten the length of the cell, and all of these are going to be ratcheting at the same time, and it's going to be this successive rowing. And right here it's attached. The only way to get it unattached is to bring in ATP, which causes it to dislodge, and then it can reach for the next one. And all of these would be doing the same thing and continuously ratcheting here. So as this breaks off, then the cross bridge forms. When the ADP leaves, then it rows, and we can have muscle contraction that way. So I introduced ATP right here. Now we've got ADP and a free P, and that's where cellular respiration comes in. We need to break down glucose in our mitochondria and take the energy from those stored molecules and bring that energy and put it into the bond of the third ATP. And we've covered all of that in cellular respiration. Now, we have calcium here. In order to get the calcium from this sarcoplasm area, this cytosol area, and back into the sarcoplasmic reticulum, we need to pump it. Okay, so that means it's active transport, and this protein, it will hold the calcium, but then we have to again use ATP, break off a phosphate in order to pump in the calcium. That same type of pumping active transport also takes place within the neuron, within the nerve cell. So we have sodium on the inside. We want to get it back